compare it to itself. Quite hard to do, right? Hard to find variation if you're only looking at one individual. So if you have a reference genome and you use other individuals to compare against it, you will find and discover more variance. Why is that? Don't overthink it. Why is that? Well, every individual is different, right? And those individual variants are going to be responsible for those differences. So if you're trying to discover variance and you compare against different people, you're going to discover more things that make those individuals unique, and those are going to be variants in the genome. So why is this? Well, most individuals are heterozygous, so 2 chromosomes from mom and dad that are not the same, which does not allow us to identify variants that differ between the parents. This tends to be quite localized, which does allow us, if you want to have broadly dispersed markers, so across the genome, so genome-wide distribution, which is what you want when trying to map traits, the best thing to do is to sample individuals that were not used for building the reference genome. And the reference individual can have at most 2 different chromosomes, right, unless it is karyotypically abnormal. So homozygosity or heterozygosity of the reference individual will therefore affect the distribution of variants across the genome. So I'll give you an example. Here we have an example of a canine reference genome. Light blue indicates homozygous regions of the genome, and dark blue indicates heterozygous regions. Now it should be noted that the individual used in this particular genome was pre-selected to be as homozygous as possible. Because when assembling a genome, it is actually helpful to have a large amount of homozygosity because it makes it easier to not be confused about the correct sequence for an area of the genome. Right? So if there's a lot of homozygosity, it's easier to put it somewhere than if there's a lot of heterozygosity when you're building the reference genome. So you can see here, light blue homozygous, dark blue heterozygous. Now, in most outbred populations, the majority should be heterozygous, and you would be able to find variants throughout the genome. However, in this example, because the individual was pre-selected for homozygosity, so it's inbred, you can see that there are a few instances where nearly the entire chromosome is homozygous. Of course, if this is the only individual, or the variants that you are trying to discover are going to be in certain regions, there will be no variants at all in other regions. Right? So you will therefore fail to discover any variants in any region for one individual that is homozygous. So to get around this, what is typically done is that we sequence other individuals from the same species, and we do something called an alignment to the reference genome to discover new variants that occur in the broader population. So what is the difference when you hear me say assembly versus an alignment? Well, an assembly has no template to follow. So imagine putting together a puzzle, but with no template to follow at all. So remember, as we did in week 2, de novo assemblies mean that no one has ever done it before. So what you are faced with is a set of genome sequencing reads that may or may not contain paired ends from the same fragment. You have no real idea of where those sequencing reads might go. In front of you is just a huge pile of sequencing reads with 3 different billion letters of DNA in little sections, and depending on your coverage, probably 30 times or more over. Right? So good luck, pile of random pieces. And here is where the assembly algorithms really shine, because they can put this together for us. As we did before, they can build you a reference genome, do all the assembly part for you. An alignment is also like doing a puzzle, but this puzzle comes with a picture on the box. It isn't an exact picture of what your puzzle is, but it is similar. You can now carefully take each piece and examine where the pattern fits in the broader picture. This makes things way easier. However, unlike a jigsaw puzzle, you're going to have anywhere from 10 to 30 copies of the pieces, depending on the coverage, which we talked about before. And some pieces will be missing entirely, especially around repeat elements. So you're basically putting a puzzle together with some type of reference picture to look at. Some pieces you're going to have 10, 20 times over, and some pieces will be missing entirely, but you still need to build the picture as accurately as possible. So to perform our alignment, we have our reference genome and our sequencing reads. We simply just look along the genome to see where they best match. So there might be a variant. In this case, the individual we are aligning has a homozygous alternate variant to the reference genome. However, sometimes this effect is caused by an error on the reference assembly, and you just have to be mindful of that, right? So here you see individual we are aligning, it does have a different variant to the reference. Now, it's very common when people first start to learn about variant discovery or variant array designs for them to presume that all variants do something. But in reality, it's highly likely that most of the variants that you will discover will be neutral. The majority arise from biochemical processes. In fact, it is often possible to tell the origin of a variant by the type of variant it is. Some arise specifically from exposure to UV, others sporadically arise during meiosis because of the ways the bases combine to replicate. The vast majority of variants do not cause phenotypic change, or at least as far as we know they don't as of today. Right? They often start off as very rare, and then they become more common due to an evolutionary process like genetic drift or selection. Right? And they may be closely selected alleles like genetic drift. Right? So while they may be enriched by genetic drift, they generally don't affect the fitness of an organism, so they can come and go with population dynamics. Right? So most variants, as far as we know, don't really do anything that affects the phenotype or affects the individual substantially. In fact, a lot of them can be lethal if they do something. So when it comes to variant genotyping and genotyping arrays are actual physical constructs. Right? So here you see an example of an Illumina genotyping array. They are glass slides that have oligonucleotide sequences annealed to them. They capture the real DNA from the organism and adhere the DNA on the slide. And the genotyping process works by analyzing the DNA that is adhered to that design site on the slide. Now, as you can imagine, in order to capture what variant or genotype the individual has at that site, you first need to know that there is a variant at that site, that there is a variant that exists at that location in the genome. You can't design something to capture to a variant that you don't even know exists. So you need to know what is in the genome, where it is, in order to design the sequence or the primer or whatever to grab it. And you actually have to physically put the sequence on the slide. So you need a sequence for each variant. So for every single loci, you'll have two different probes that can grab that, depending on what variant is at that location. Now what that means is that variants, the number of variants you can put on the slide are going to depend on if those locations are polymorphic and if they have more than two alleles at that location, because it's going to take another probe, right? So it's a physical space that is being occupied, and that is going to, of course, restrict the number of variants that you can identify. So this picture is an example from Illumina. So it's zoomed in on an array. You can see that the probes that are designed to attach if they match the DNA sequence. So C probes light up green, the alternative probe lights up red. And again, the thing to be aware of is that when a probe doesn't work, it is possible that you get very low intensity of both probes as a result of cross-reaction from nearby reads, which would falsely call something as heterozygous. Right? So there are some DNA quality measures that must be accounted for when you're looking at genotyping arrays as well. Now there are multiple applications for genotyping arrays. Some are listed here. You can map genes associated with phenotypes, you can characterize populations, you can observe evolutionary processes, you can monitor diversity, and you can assess relationships among individuals. Now, those are genotyping arrays. They're extremely common, I think, in the animal space, especially on the wildlife side, not so much with, I think, more endangered, more domestic programs, but heavily used on the livestock side. I don't think you guys have genotyping arrays in the bees? Yeah. So and the reason for that is it takes quite a lot of investment and funding to build genotyping arrays, but once you've done that, they're extremely cost efficient to use. So you can put 96 individuals usually on a plate, and the beef industry and the dairy industry might put like 100,000, 200,000 variants per animal, over 100,000 animals a year. So it's extremely cost efficient once you've built them, and you can get, depending on the array, 500,000 markers evenly distributed across the genome rather than sequencing. As you guys are getting an appreciation for here, there's a lot that goes into the sequencing. It's not just generating the data. The processing and analyses take a lot of time and resources, and arrays are kind of a way to shortcut that. So those are genotyping arrays, which have been the predominant method for the last 10 to 15 years. But as genome sequencing has gotten cheaper, additional genotyping strategies have been developed. So all strategies have strengths and weaknesses. And alternative strategies are popular in biology and environmental sequencing applications because they are fabulous for conservation and evolutionary applications. And the reason for this is some of these platforms, you don't actually need to know where the variants are, but you want to know the amount of variant sharing between individuals, between individual populations. And here you see low-pass sequencing with imputation, RAT-seq, so restriction site associated DNA markers, and DART-seq, which I've seen used in species that don't have very well-built reference genomes, so I think it's used quite a lot in wildlife, reduced representation sequencing. So you'll also find when it comes to sequencing and genotyping arrays, what one industry or one discipline or one species considers big data, another one not so much. So I'll give you a best example I can give, of course, is going to be horse. In the equine area, we have multiple genotyping arrays. One of them is, the old one is 50,000 markers. Keeping in mind the horse genome is 2.7 billion bases. There's a new one that's 80,000 markers. There's another one that's 670,000 markers. And there's another that's 2 million markers, right? All genotyping arrays. So those can be used for a variety of tasks, a lot of mapping traits, we use them for diversity. In dog, I think it's about 250,000, okay? But when I went to look at DART sequencing, because we were like, cost efficiency, you're doing research, right? Budget's always an issue. And I said, well, how many markers can I get? Because for the high density horse array, for $100, $125, I can get over 500,000 markers on a horse, per horse. So a couple $100, over 500,000 markers per horse. But if I use the DART sequencing, it's a little cheaper. I think it's about $100 an animal, but they can only guarantee me 20,000 markers, right? Because it's low pass and it's cheaper, but you don't get as much information. Well, 20,000 would be more than enough for lots of wildlife species, but for domestic species, it would not be considered a robust analysis at all. Right? So depending on what you're doing, so human stuff, think about coverage of a genome. For the animals, 15x, maybe 30x, that's more than enough, but for human, 100, 200x per individual. So the species you work with is going to impact the quality control and the amount of data you can generate as well. So genotyping arrays, as I mentioned, are very expensive to design, but they are highly accurate and much cheaper once they are designed. In fact, they're actually sometimes better at certain parts of the genome, especially around centromeres and things like that, repetitive elements, than genome sequencing itself. Right? So new technologies are way cheaper, and there are various methods used to make prices comparable to purchasing a genotyping array after it is constructed. However, other strategies don't get cheaper as you go. So they simply ask you to sequence less, so reduce genome complexity. So while there are some other options that might be more cost efficient, if you do more individuals, it doesn't get cheaper. Versus the arrays, the more that you do, the cheaper it gets. So I think for the beef cattle and the dairy cattle, they do so many a year, they might be down to $75 or less per individual. So in order to do variant discovery, you need a few ingredients to do it. Ideally, a reference genome, so you can tell precisely where markers are located and ensure that the marker dispersion is even across the genome that you're interested in, right? Because if you're trying to map a trait, but all you've done is pick markers that are on chromosome 2 and 3, what if the causative mutation, what if what you're interested in is on chromosome 12 or 15, right? So you want evenly distributed markers when you're looking at variants. You need sequencing reads from different individuals than your reference. So you have the ability to align these reads to your reference. And you want, of course, bioinformatics tools that perform the genome alignment of the reads within the reference genome. and another tool to analyze the alignment to ascertain whether there is a variant there or not, and how high quality that variant is. If you just see one random variant pop up, how likely are you to believe it if you see it in 1 in 1,000 individuals? Well, depends on the quality control, right? Because it could be, if you're sequencing 2.7 billion nucleotides, there's bound to be some random errors in there. So usually you want to see things a few times to believe that it isn't truly a new variant, especially if it's something that you think might be associated with a disease or disorder. So these are the processes that we're going to kind of step through today and this morning's, the rest of the morning's activities. Now remember, genetic variants are the DNA alterations that make us an individual, as well as being able to help us understand how evolution has acted upon the species to make them different. However, the world of genomics is still in its infancy with respect to our understanding of the effects of genetic variation on phenotype outside of the protein coding sequences. So before we can understand the impacts of the genetic variants have, we first need to identify them, sort those that are normal, so they have no real effect, they're erroneous variants, from those that are important. And then of course, design tools that help us to test our theories about the impacts of the variants that we have already identified in many new situations. And we prefer to all do all this at a low cost, right? So if you recall from earlier, markers discovered in a genome assembly are often patchy, with some chromosomes showing few variants. Of course, microorganisms, something to keep in mind for today's activity, only have a single chromosome and thus cannot be heterozygous. For this reason, marker discovery using a single microorganism or microorganism colony may not be useful. However, for demonstration purposes in today's workshop, you will be using reads and reference from the quote-unquote same microorganism just to see what you come up with, right? It is possible that reads and references may be slightly different strains and may therefore be diverged in evolution. So don't assume that you'll find nothing even though it is the same species, right? They could be slightly diverged, you might find something. And remember, the more diverged two organisms are, the more SNVs and indels you will find when you compare them. So what you are doing today is what you will also be done or would be done for any other mammal without a reference genome, right? So you would use your own reference assembly and then you'd align the sequencing reads back to it to try to identify variants. So with that, go ahead and start your virtual machines back up. And while they are starting up, I will remind you, right, that patience and perseverance has a magical effect before which difficulties disappear and obstacles vanish. So hopefully you're seeing that a lot of the mistakes that you see, sometimes it's just something a simple typo. So never assume, and I saw some really good practice, really good habits earlier. Never assume that the file that you have created is what you think it is. It's always good to head it or have a look to see that it's what you think it is. Never assume that, oh, the input file has the right name, it must be the right file. What is in that file? What's the format supposed to be, right? Because sometimes it's something so small that you don't realize it because you're overlooking it. And I know that my own students heard me complaining this earlier this week because make sure you guys listen, I spent an hour trying to solve a problem that was really making me want to rip my own computer out the wall. And it turns out I had created a new file and just wasn't looking at the right file. Like I just kept opening the same file thinking that the code wasn't working. It was working perfectly and it took me an hour to see that I was like, oh, wrong file, my bad. Right? So we understand the frustration, it's just part of it. I also want to encourage you, I heard a lot of great questions earlier here, like, oh, what would happen if I did this thing, if I changed this threshold? You can change the thresholds as you want. If you've done it already and you have the answer and you're just waiting on your classmates to kind of catch up, we're helping other people, change one of the thresholds and see what happens, right? See how does it affect your input? How does it affect the output? If it gives you an error, okay, then that's something you probably shouldn't change, right? So don't be afraid to experiment a little bit with the coding, especially when it comes to the quality control, because it will help your understanding of how that quality control impacts what you were doing. So while the machines are starting up, we'll give you about a 2 or 3 minute break. Don't get too loud or don't wander off because we will pick back up in about 3 minutes and pick up for the next activity.